Assalamualaikum everyone, this is Zohaib Tahir from ZMeds and today we are going to talk about cholinergic drugs and neurons. First of all, we will see cholinergic and adrenergic drugs. Those drugs which are going to stimulate cholinergic receptors are going to inhibit the cholinergic receptors are known as cholinergic drugs, while those drugs which are going to inhibit adrenergic receptors or stimulate adrenergic receptors are known as adrenergic drugs. So there will be both kind of actions, stimulatory or inhibitory. If the drug is going to stimulate any kind of action on by acting on the adrenergic or cholinergic receptors, then it is known as agonist. While blocking the action will give it a term antagonist. Next, we have to see neurotransmitter acetylcholine that will be released at three different sites in human body in central nervous system and peripheral nervous system. Number one, preganglionic fibers of both sympathetic and parasympathetic nervous system are going to release acetylcholine. So in case of all kinds of nerve impulse conduction, acetylcholine is used. While in case of postganglionic fibers, all parasympathetic plus sweat glands plus adrenal medulla will have the innervation from acetylcholine. Or postganglion fibers of just parasympathetic sweat glands and adrenal medulla will release acetylcholine to bind with the nicotinic or cholinergic receptors or muscarinic receptors. While in case of somatic nervous system, there will be an N type of nicotinic receptors present at neuromuscular junctions of the muscle where acetylcholine will be attached. After that, we have to see different steps in the neurotransmission that includes synthesis of acetylcholine, after that storage into the vesicles, then release of neurotransmitter acetylcholine from the vesicle into the synaptic cleft, then binding of acetylcholine with its receptors, and then degradation after being utilization, and then recycling of degraded parts of acetylcholine. After that, we have the very initial step known as synthesis, in which choline along with sodium will be co-transported into the neuron, and that step can be blocked by hemicholinum. At that very site, choline along with acetyl-CoA is transformed into acetylcholine with the help of an enzyme known as choline acetyl transferase. After that, we have the second step of storage, in which acetylcholine is stored in the vesicles along with ATP. So the storage vesicle or the coat around the neurotransmitter is going to be made up of proteoglycan, and there will be co-transmission, as we have seen in the previous step of synthesis, in which sodium and choline were co-transported. Sodium and choline were moving both into the neuron. Here in this case, ATP and choline both are moving into the vesicle. After that, we have a release of neurotransmitter acetylcholine, in which you can see action membrane potential is generated with the help of sodium channels, and sodium channels causes the calcium channels to open. And when calcium channels open, what will happen? There will be movement of the vesicle from inside of the neuron to one end of the neuron, and as a result, there will be fusion of neurotransmitter containing a vesicle with the membrane of the neuron. So as a result, there will be release of neurotransmitter into the synaptic cleft. Well, this release can be inhibited by botulinum, and black widow venom causes massive increase or release of acetylcholine. After that, we have to discuss the binding phenomena, in which acetylcholine binds with its nicotinic or muscarinic receptors. So they are going to stimulate muscarinic or nicotinic receptors accordingly wherever the site is. Fifth step is of degradation and recycling. Well, acetylcholine esterase is an enzyme which breaks the acetylcholine into acetyl or acetate group and choline. So choline is reabsorbed and reutilized to get acetylcholine again. Here is the diagram in which you can see choline and sodium are co-transported into the neuron, and then with the help of an enzyme known as acetyltransferase, there will be formation of acetylcholine from acetyl coenzyme A and choline. After that, acetylcholine will be shifted into the vesicle, and then upon proper stimulation from the calcium, vesicle will move towards presynaptic membrane or presynaptic knob, where it will bind or will fuse with presynaptic knob, presynaptic membrane, and there will be release of neurotransmitter. After the release of neurotransmitter into synaptic cleft, there are muscarinic or nicotinic receptors present, and drug or acetylcholine or the ligand will bind to the receptor, resulting in the generation of secondary messenger or opening of the voltage gated channels. And after that, acetylcholine will be taken up and degraded with the help of an enzyme acetylcholine esterase. The degraded form includes choline and acetate or acetyl, and these both can be reutilized to get acetylcholine again. After that, we have cholinergic agonists, where there are direct acting cholinergic agonists which include acetylcholine, bethanycol, carbachol, nicotine, pilocarpine, methacholine, while indirect direct acting have two categories. Number one is reversible and number two irreversible. Reversible includes edrophonium, neostigmine, physostigmine, pyridostigmine, while in case of irreversible, we have ecothiophate and organophosphates. The poisoning of organophosphate and ecothiophate can be reversed by adding a drug known as pralidoxime. That is a reactivator. Reactivator means acetylcholinesterase enzyme is actually getting phosphorylated from these drugs known as ecothiophate and organophosphate. Pyridoxime vanishes the phosphate linkage of these drugs with cholinesterase, and then cholinesterase is ready to be utilized. If you have any questions related to this topic, then you can ask in the comment section below. And to get very unique concepts about cholinergic stimulation, parasympathetic and sympathetic nervous systems, you have to follow the lecture about Introduction to Autonomic Nervous System Part 1, Part 2 and Part 3. Links of all those videos are present in the description box below. Hopefully you have liked the video. If you haven't, then let me know in the comment section below. I will remove your doubts. Till then, it's Zohaib Tahir from ZMeds signing off.